Actually, I'm cheating today. I'm going to talk about two different things. In the first half, I'm going to talk about some population level genomic and genetic testing we did to try and understand kind of the scope of people we don't know about with ALD. And in the second half, I'm going to try and open up kind of a new exploration for the role of ABCD1. I think there's a lot of great work that we'll be hearing about in part today from Stefan Kemp about understanding the contribution of very long-chain fatty acids and an improved diagnostic biomarker with C26, but also I want to explore a little bit in the second half of the talk just if there's a different role for ABCD1 in cholesterol trafficking in the cell. So various disclosures, nothing really related to what I'm talking about today. And then just thanks to some of the different supporters for our work, really doing the basic science that we're talking about is hugely expensive, and so we're really fortunate to have a lot of great supporters, some of whom are in the room today. So there's two different teams that really contributed to the work. The group on the left, Stacy and Bryce, helped us with the population level genomic testing, and then the folks on the right are in our lab, Courtney and Tammy, who did a lot of the work that I'll be showing in the second half of the talk. So in the first half, I'm going to talk a little bit about trying to understand where are the ALD patients. So some of them, of course, are in this room, so we're happy that you guys are here. But what we're seeing now with newborn screening is that there's a lot of people out there we don't know about. And so using genomic testing in a healthy population, our goal is to try and understand who are the patients out there that we don't know about, what symptoms do they have, are there things that we're missing. And so the way, there's been various different estimates of the population incidence of ALD, ranging anywhere from 1 in 10,000 to 1 in 55,000 live births. This comes from a variety of different estimation methods, ranging from newborn screening, which has really kind of taken off in the past few years, using estimates based on different kind of quantifications of genomic incidence, different center studies, and so the number is quite large. And so really our question was, is there kind of a healthy cohort of ALD patients we don't know about, or a cohort that's out there that has different symptoms than what we're expecting? This also relates to kind of our understanding of what are the contributions of these variants that we're trying to understand with their, which variants are pathogenic, which variants aren't pathogenic, as well as how penetrant different variants are. So meaning basically, if you have one of these variants or mutations, how likely are you to have symptoms? So there's a kind of a variety of causes that could be leading to this. So for example, maybe you just have a later age of onset, so you're not showing up to the healthcare system until you're older. Of course, the disease may have symptoms that are milder or aren't bringing you to the doctor's office. You may have different symptoms than what caused people to be testing for adrenoleukodystrophy, or there may be genetic modifiers in the population that change how your symptoms are presenting. Alternatively, maybe we're doing a great job and we just start missing patients. So the way we did this project is we were part of a large study in the Intermountain West, mostly in Utah, called the Heredogene Population Study. This ran over 3 years. It's a collaboration of Intermountain Healthcare, which we're part of, which is the large healthcare system in the Intermountain West, with Decode Genetics, which is based in Iceland, and Amgen. It was enrollment was across the entire Intermountain West region in the United States, so a very large geographic area. And it was a combination of whole genome sequencing of patients as well as what's called imputation. And imputation is basically a way to do kind of sparse testing across a person's genome and then impute or predict what the rest of the variants are. It's not quite as comprehensive as a whole genome sequencing, but much cheaper, much faster. There was return of results to patients if they had an actual genetic result. However, it was a population-based screening, so it wasn't for diagnosis, and so it was basically kind of a healthy population screen of folks. And just as kind of a note for the results I'm going to talk about, at the same time that this study was going on, this hereditary study, we had just opened newborn screening in Utah in the late fall of 2020, so really 2021 is when we were starting to get rolling, and so we were just starting to pick up larger parents of babies and grandparents who didn't realize that they had ALD in the family. So we had a total of 245,000 participants in the study. This included 34,000 people who had whole genome sequencing, and another 210,000 people who had imputation, so a very large swath of people that were enrolled. And so these are the, so we went back and looked at the data. So we looked at not only ABCD1 for ALD, but we looked at multiple different causes of leukodystrophy because I wanted to look at a whole bunch of different leukodystrophy genes. And just to kind of point out here in the bottom row, I kind of blew up this thing. We looked at approximately 800 different variants. This is not every variant that's known in the ABCD1 gene, but we limit ourselves to variants that are listed in ClinVar, which is kind of a standard cataloging of genetic mutations or variants in clinical genetics as a way to kind of standardize how we did this across all the different genes. So this is the results. So the top is for all the different genes, but then in the bottom here, I've just blown it up for ABCD1. And basically, in this 240,000 people cohort, we saw no one who had one of these 800 plus variants in ABCD1. So this is the answer, 0, 0, 0, 0 across the row here. So this is kind of surprising. This is kind of a contrast example. So for a different gene called CSF1R, which is an adult onset leukodystrophy, which I'm just using as a comparison here, we saw 2 people who didn't realize they had the condition and had kind of symptoms that you may or may not relate to having this gene variant. So on the one hand, we didn't see anyone in this entire cohort of 240,000 people who had ABCD1 variants or ALD, but we did pick up other people with different leukodystrophies in our cohort. So this is kind of surprising. So basically, we're not missing people in this large, healthy cohort of adult ALD patients, at least with the 800 variants that we looked at. So it could be, of course, that people who signed up for this study, that they're kind of self-selecting to be in it or not to be in it. We didn't have every single variant included in our analysis just because we limit ourselves to ClinVar variants, but still it's 800 different variants, most of which are well known in the ALD literature. So it seems like patients who are symptomatic seem to be symptomatic enough that they're kind of self-excluding from being in a healthy cohort, suggesting that at least at the level at a population level, that ALD is penetrant enough to have symptoms that people recognize. Also, it's a little bit unclear, but it seems like we're all worried about these VUS variants that we're picking up in newborn screening, and those seem to be not showing up in our cohort. So it was kind of surprising at this large population level not to pick up ABCD1 variants, and so we're kind of digging into this further, but it's so interesting for ABCD1, and then of course interesting for these other leukodystrophies where we are picking up patients who don't realize that they have these genetic changes. So I'm going to stop there for this first part of the talk. And happy to take questions now or also at the end.
One question for you. How do you think the newborn screening is going to change our understanding of the data that seems like a gold standard of.
So I'll repeat the question. So the question is, I'll try and restate it, is how is the newborn screening kind of affecting our understanding of the incidence rate? So I'll use Utah as an example. So in Utah, prior to 2020, we were following about maybe 6 families with ALD. Now we're following about 80 to 100 families, and in most of those families, of course, we picked up the baby, but then we picked up, parents and grandparents, so it's like totally changed our understanding. However, in many of these families, we're running into this issue where many of the variants that are identified in these families have never been either reported before, and then in these extended families, there's no history of anyone with any disease at all, or very mild symptoms. And so it's been very confusing to try and predict risk or kind of counsel families what level of anxiety they should have. So on the one hand, the incidence level kind of matches what's been reported at a population level, but on the other hand, how to help families in the long run continues to be not totally clear. So I'll transition now to talk about cholesterol pathways in ALD. And so really this is work that's still very much in progress, and it's a kind of avenue of research that we have looked at because we have been wondering whether or not there's a role for ABCD1 that we're not understanding with our focus on the kind of involvement of ALD in very long-chain fatty acid metabolism. So as we kind of have been discussing, the relationship from different individual mutations to the phenotype is still being kind of worked out. So with any given new mutation, it's not always possible when the mutation shows up to say, what's the phenotype that we can expect. Further, as we've kind of been looking at the VLCFA levels, don't correlate necessarily to what the symptoms are or to the severity of disease or to which type of disease that someone's going to have. There's a lot of hope, and I think our next speaker will be really convincing us that C26 lyso-Pc is the way to go, but I think that there's still more work to do there to make sure that this all pans out. So really, what we're trying to do in this work is try to understand how does ABC1 contributing to the disease that we see in the spinal cord and in the brain, in the adrenal cortex. And so really, we know that ABC1 works as a transporter for very long-chain fatty acids, but then how does that translate then into these different symptoms that we're seeing, the neuropathy symptoms, the adrenal insufficiency, and the inflammation in cerebral ALD. And so a couple years ago now, we did a multi-platform screen in zebrafish where we really banked on the fact that you can work with thousands of animals because they're small and inexpensive, and do a lot of experimental manipulation and do this kind of at scale. And so we basically compared wild-type zebrafish to mutant zebrafish and did a variety of interventions, including intervening with effects on cholesterol ester levels as well as on inflammation in the brain. And we then did kind of two different sets of tests. We did wild-type animals compared to mutant animals. And we also compared animals with or without intervention, whether it was with or without drug or with or without the inflammation. We then did transcriptomics, so doing RNA-seq. We did lipidomics, and that was in collaboration with Stefan Kemp's group. And then analyzed all this data to kind of see what patterns we saw coming out of the data. And so with the different sets of data, first in the transcriptomics, we saw a lot of dysregulation of cholesterol biosynthesis genes. So this was something that's been reported before, but kind of confirming to us that cholesterol is involved in these pathways critical to ALD. And in the lipidomics, we saw, in addition to the kind of the known accumulation of very long-chain fatty acids, we saw that cholesterol ester levels were kind of a key factor that was not only change in wild-type versus mutants, but was affected by our drug intervention, and was also affected by the with the wild-type or mutant status of the animals. So this really kind of emphasized to us that, were we missing looking at cholesterol pathways in ALD? The next thing is that kind of looking back through kind of the literature, the involvement of cholesterol has been known for a long time, and so cholesterol esters accumulate in the brain of patients with ALD And this also has been seen in cerebral ALD. There's many papers now looking at cholesterol metabolism and seeing that how the ALD patient cells control cholesterol is different in patients. And then there's also groups now looking at how cholesterol accumulates in the different parts of the cell, for example, the lysosomes of people with cholesterol. In kind of a population level study we did a couple of years ago now, we looked in adult AMN patients, and so we were trying to understand what the pattern of disease is in adult patients with AMN, and many of them had kind of disease phenotypes that you'd expect with people with lipid abnormalities. So for example, we saw high rates of liver disease, of peripheral vascular disease, of cerebrovascular disease, and congestive heart failure in the AMN population. And so that really suggested that is there like a level of lipid dysfunction in AMN and ALD that we haven't looked at. The final piece I wanted to kind of set up as we kind of look at the story is that we looked at something called evolutionary rate covariation. So I'm just going to call it ERC. It's a little bit easier to say. And so this was a project we did a couple years ago where we looked at changes in genes across evolutionary time. And so the concept of it is that genes that work together are going to change in evolutionary time similarly, because if one gene goes, if they have to work together and one gene goes off in one direction, and its partner doesn't move in that same direction, it's not going to be able to work together. And so genes that have high rates of covariation must be doing something functionally similar to each other. And so this was looking at ABCD1, and it's a little bit complicated, but basically ABCD1 is shown here at the bottom in the kind of this blue box, and genes that that across evolutionary time show this covariation are marked in red. And so we saw about close to 1,000 genes where they had a high ERC, meaning that they kind of covariated together across evolutionary time, as well as were statistically significant. And we saw an overrepresentation of genes involved in cholesterol metabolism, synthesis, and transport. So again, kind of really helping us focus on the cholesterol pathways. So typical cholesterol pathway, just in a cell kind of at large, is cholesterol is taken up at the cellular membrane. It's transported from lysosomes to peroxisomes to the endoplasmic reticulum, and then ultimately can control how biosynthesis is handled in the nucleus. And there's been a group that in 2015 suggested, whoops, if I can go backwards here, that ABCD1 is necessary for cholesterol trafficking. So basically, they did a knockout of cholesterol in cells they did a knockout of ABCD1 in cells and saw a disruption of cholesterol trafficking. And so our approach has been to use patient cells, generate a kind of a comprehensive battery of staining for different aspects of cholesterol trafficking, do imaging, and then do the quantification. And really that's been work that Courtney's been doing. And looking at different kind of metrics in the cells. So we have measures in the lysosome and the peroxisome, and something called lysosome and peroxisome membrane contact, looking at the peroxisomes, both quantifying peroxisomes as well as what their cholesterol content is, quantifying lysosomes and what their cholesterol content is, and then looking at components of the peroxisome and how the cholesterol is trafficked across the, what kind of molecular components are actually responsible for the trafficking. And so this is fairly detailed work where using various quantification methods, so for example, using staining for lysosomes and peroxisomes, shown here in red and green, and then the areas of overlap are shown here by using pixel quantification, where you take the overlap and then kind of subtract out what's remaining where the areas of overlap are for the lysosome to peroxisome contact. The same thing here, where we then kind of zoom in to see where the contact is happening and actually looking at the membrane contact between the lysosomes and peroxisomes. And then quantifying in the peroxisomes and lysosomes what the cholesterol content is and how it's affected by different kinds of manipulation. Sorry, this is kind of slow here. So for all these previous slides, we've all been doing kind of single or double stains, and actually it ends up being quite a lot of work to try and get data from that. And so Courtney has figured out basically a quad stain, so being able to visualize peroxisomes, lysosomes, and cholesterol at the same time. And then finally, by tagging ABCD1, we can manipulate what's happening in the cells by adding wild type or mutant ABCD1 and seeing how that affects the cholesterol trafficking. And so really our goal is hopefully as a teaser for next year, where we're trying to see is ABCD1 not just involved in trafficking very long-chain fatty acids and controlling VLCFA levels, but whether ABC1 is necessary for normal cholesterol trafficking, and if that derives disease phenotype. We're looking both in wild-type and mutants, as well as with different kinds of manipulation, including drugs and different kind of genetic mutations. So I wanted to thank all the people who did this work. It's been a lot of work, especially by Tammy, our lab scientist, and Courtney, our graduate student. And Courtney was, I think, here last year and has been supported by ALD Connect with the Emerging Investigator Grant. So thanks so much for your attention, and happy to take any questions.
Hi, Josh. When you were looking at AMN men who presented with high levels of cholesterol, were you factoring in lifestyle choices, diet, exercise?
No, so good question. So the question was related to that study in 2024 in Orphanet, where we looked at the rates of different health conditions in adult men with AMN. And so it was not controlled for, like as an example, maybe if you have AMN, then it causes you to exercise less, which then kind of contributes to having peripheral vascular disease. So there wasn't really a way to control for that. And so it could be all secondary effects for sure, so we don't know.
Wonderful work, Josh. I was just curious. I mean, you're talking about cholesterol synthesis, and the graphs showing the evolutionary correlation here with cholesterol genes. It's just fascinating. It strikes me that there's a very organ-specific component here, and vulnerability of organs that are particularly rich in cholesterol uptake. And that might be a separate thing than just cholesterol synthesis. And so thinking of adrenal cortex, thinking of the nervous system, how do you think you can interrogate that specific tissue cell type? Or maybe I missed it. Is that still coming through in the studies you're proposing?
That's a good point, Florian. So one concern, so as you may have noticed, we did some whole animal studies in zebrafish, but then for the trafficking studies, we're using patient fibroblasts for kind of experimental ease. Fibroblasts are not people, and so we've been concerned a little bit about a couple of things. One is related to your question, like, is it different in fibroblasts versus adrenal cortex cells? And that then has taken us down a rabbit hole of whether or not we need male adrenal cortex cells or do we need female adrenal cortex cells. And so actually it turns out that most adrenal cortex cells are female derived, interestingly. And then we're also worried a little bit because the cholesterol and lipid pathways are such a quagmire that different kinds of media that you use for doing this experimentally just affect the results as well. So there's a bunch of experimental problems that we are worrying about. But yeah, good point.
Other questions? Great. Thanks so much, Josh. That was excellent.